Hello, my lovely anatomists. Michele Glass here, and we are still looking at the anatomy of the muscle fiber. Remember, fiber here, we're translating to cell. And we see our diagram from the OpenStax textbook of our muscle fiber. So remember, our muscle fiber is called a fiber because it's cylindrical in length and super duper long, is multinucleated, has lots of nuclei. We call the cell membrane the sarcolemma. We see mitochondria here, just like we had in our cells in chapter 3. And then we see that the muscle fiber is made up of a bunch of smaller cylindrical shaped structures that we call a myofibril. And the key feature of a myofibril is that it's containing thin filaments, which are primarily actin, and thick filaments, which are primarily composed of myosin. But then we see that the myofibril is organized in a unit called a sarcomere. And so that's where we need to spend a little bit more of our time kind of figuring out that particular shape. So when we talk about our sarcomere unit, we should think about this as the smallest functional unit of the muscle fiber. This is where all of the contraction business is taking place. And remember that the sarcomere is located on the myofibril. And the myofibril, remember, is cylindrical in shape, and it's the full length of the muscle fiber, the muscle cell. And so in this picture of the sarcomere that we have here, we're looking at one side of that myofibril. So instead of seeing the whole 3D structure here, we're seeing a very flat image. Now we've talked about the myofibril and thus the sarcomere as being composed of the thick filament and the thin filament. So we see that the sarcomere and thus the myofibril are composed of the thick filament and the thin filament. And what we're talking about here with these filaments, we're talking about our contractile proteins. We can also talk about them as myofilaments, where the myo prefix there is talking about muscle. So it's basically saying proteins of the muscle, right? Now let's take a look first at our thin filament, and we'll see that it's composed primarily of actin. But we see also these proteins troponin and tropomyosin in play. So taking a look at our actin, our thin filament, actin is typically shown as looking like beads on a strand. And we see that it's 2 strands of beads and they're kind of wrapped around each other. So the actin would be in this picture like the bead. And we see that there is what's described as a binding site for myosin on that bead. There's a binding site for myosin on that bead. But sitting in the way of that binding site, we have what's called troponin and tropomyosin. And I often talk about this as the troponin tropomyosin complex, which helps me to remember that the troponin is going to be kind of like the outermost, and it's going to hold the tropomyosin in place. So the troponin is going to be holding the tropomyosin in place. And so then this little ribbon of protein that we see is the tropomyosin. And it's going to actually cover the myosin binding site. So you can think of the tropomyosin as like a ribbon that's sitting on top of the active site or the binding site on your actin. And the troponin is holding that in place. and this is at rest. So when the muscle is at rest, you have this actin, which looks like a beaded necklace, and you have these special kind of parts of the bead that's called the myosin binding site. And at rest, you have this ribbon, tryptomyosin, sitting on top of that binding site, and the troponin is kind of clipping or holding that in place. This actin, troponin, tryptomyosin complex is all part of the thin filament. All part of the thin filament. If we look instead then at the thick filament, we see that it is composed primarily of the protein myosin. And the myosin looks somewhat like a golf club. So you have like this long myosin tail, and you have sort of this flexible bendy neck, and then you have these heads that look sort of like the golf club part. Now the heads have two important pieces. The heads have two important pieces. So the head is going to have both an actin binding site, and the actin binding site is going to be, you know, really in love with that myosin binding site. So here you see there's a connection. So remember that in order to get contraction, you have to have a connection of the actin and the myosin. And where do they connect? Well, the actin binding site on the myosin head is attracted to the myosin binding site on the actin bead. And then we have an ATP binding site. So this is the other important part of the myosin head. And this actually has an ATP ACE. ASE ending tells us this is an enzyme. So this is an enzyme that breaks down ATP. This is an enzyme that breaks down ATP. Remember, we've mentioned that muscle contraction is a high ATP demanding process, a highly energetic process. And so what we'll see is those heads will actually bind with ATP and break down each of those ATP molecules. Now, when you look at the thick filament, it's important to remember that it's a 3D structure. So both the thin and the thick filaments are 3D structures. And so our thick filament looks sort of like a cylindrical shaped bundle with these heads sticking out all the way in all the different directions. Okay. Let's go back up to our idea of a sarcomere, and let's see if we can figure this out a little bit. So the sarcomere, remember, is our smallest functional unit of the muscle. And this is how your thin and your thick filaments are organized on that myofibril. This is how your thin and your thick filaments are organized on your myofibril. Okay. And what we can see is the border of our sarcomere is going to be what we describe as the Z line. And the Z line looks like a bunch of Z's together. It's got this squiggly line. So a single sarcomere unit extends from a Z line to Z line. Now we're seeing in this diagram that the thin filament is represented with the green color. So we see that the Z line is in part composed of that thin filament. And then we see exactly extending off of the Z line, the thin filament shown in green. And we see that the thin filament then is extending in both directions, left and right sides of that Z line. And of course, this is 3D structure, so all the way around. So we have this area called the I band. This diagram calls it the lighter I band. We can just call it I band. The I band is going to have just thin filaments. And it goes across Z lines. It includes the Z line. So it's the Z line and then the thin filaments on either side. I use the memory device Itsy Bitsy, as in like the Itsy Bitsy spider. Okay, so I band Itsy Bitsy thin filaments. If you look in the very center, of your sarcomere, you have an M line. So the M line is the center line. And notice it's made up of thick filament. And then extending on either side of the M line, you have the thick filaments. You have the thick filament on either side of the M line. And you have an area that's called the H band or H zone here. And so if you pay attention to the H zone, it has just thick filaments. And it includes the M line. The H bone zone has just thick filaments and it includes the M line. So I like to talk about the H band or H zone as heavy for thick filament. Now on this diagram, they're not showing you what would be called the zone of overlap. The zone of overlap would be where you have thick and thin filaments together overlapping. So that's a super creative name, right? That's a super easy for us to understand name. The last thing for us to look at is what's called the A band. And so when we look at the A band, what we should see is it's the whole area that's thick filament. So it includes a little bit of the overlap. It includes the H zone. It includes the M line. So this area that's the M line, the H zone, zone of overlap, all of that is basically the A band. And with the A band, I like to think about that as always the same. My picture moved. There we go. So I like to think of my A band as always the same, meaning when the muscle cell is at rest or when the muscle cell is contracted, the A band doesn't change in size. So that'll be significant as we move through. Let's take a look at this previous picture here. Looking here at this sarcomere, we may see a few different sections of terms. So choose red to label. So we have our sarcomere unit and we see that it's bordered Z disc to Z disc or Z line to Z line. So that's how we know what is a sarcomere. And here we're seeing it as our 3D cylindrical shaped myofibril. So that's helping us have a better understanding. And we can see that the myofibril then is a sarcomere unit, sarcomere unit, sarcomere unit, sarcomere unit, all the way down the line. Now at the very center of your sarcomere is your M line. And so we see that actually not labeled within the sarcomere label, but kind of to the side. And then we're seeing here kind of that purplish color representing those thick filaments. So we have here what's called the H zone, the heavy zone, where that's just thick filament, M line, thick filament. So there's no thin filaments represented in the H zone. When we want to talk about just the thin filaments, we look for the I band. So that's going to be thin filament, Z line, thin filament. Remember here, I'm using my memory device, Itsy Bitsy, to help myself remember that. And then we have what's called the A band. And notice that the A band goes from the thick filament, zone of overlap, H zone, M line, H zone, overlap, and so forth. So the A band is pretty much this whole space where you have the thick filament. And then let's make sure that we've labeled our overlap zone. So you can call it zone of overlap or overlap zone, either way you want to do that. Now, all of this is important because this helps us to see how the muscle contracts and changes during contraction, I should say. So this top picture is at rest. And what we see here is the dark green Z line, and then we see the thin filament coming off the Z line. And so we see that as our I band. And then we start getting into where we can see a zone of overlap. And so in the zone of overlap, you have a thin filament, thin, thick, thin, thick, thin. And so if we're getting the thick filament, we can trace from one thick filament all the way across the other side. That's that A band. We have at the very center of the sarcomere unit that M line, and we can see there's a little bit of thick filament on either side. So that's our H zone, our heavy zone. So this is our resting sarcomere here at the top, where we see these black lines, these black arrows on each side of the sarcomere. We're seeing that's the force, that's the contraction that's happening. And what we should see is that the myosin is going to pull the actin toward the M line. So the myosin is going to pull the actin toward the M line. So what we're seeing is the actin, the thin filaments are coming closer and closer and closer and closer to the M line. So when this happens, we're increasing our area of overlap. So there's more overlap between the thin and thick filament. We see that we have reduced the H zone. So pretty much we've got those actin, those thin filaments right up there close to the M line, and there's no space on either side of the M line. We've also reduced our I band, our area that's just thin filaments, because those thin filaments get pulled across the thick, and so we have less of that I band. So we've increased our zone of overlap. We've reduced both our H zone and our I band, right? And then what's staying the same is the A band doesn't change. And so this is how we know that when the myosin pulls the actin towards the M line, the thin and the thick filaments slide across each other. There's lots of great animations showing the sliding filament theory of muscle contraction. So that's a great thing for you to look up on YouTube if you want to get more of an animated version of this. That's it for our anatomical studies. We're ready to move more fully into physiology with our next video. So stay tuned. As always, take care of yourselves and each other.